Hi everyone, if you haven't already watched the Edpuzzle video about DNA's discovery, please make sure you stop this lecture right now and go back and watch that Edpuzzle video, as I will be making references to it as we go through today's notes. Right. So I want to talk to you a little bit about DNA before we go into its function. So let's look at its structure first. Make sure that you have a piece of paper and you're writing down some notes as I go through this material. So DNA stands for deoxyribonucleic acid. If you look at that word deoxyribonucleic acid, you can figure out what type of organic molecule it is. So think back to when we learned about proteins, lipids, nucleic acids, and carbohydrates. So within its name, we know that it's a nucleic acid. The second thing I want you to notice is at the beginning of the word, deoxyribo is a clue as to what type of sugar is found in its nucleotide or building block. So DNA, or deoxyribonucleic acid, is made up of nucleotides that contain the sugar deoxyribose. And if you recall from when we talked about organic molecules, DNA is the genetic material that's passed on from one generation of cells to another, or from one generation of organisms to another. And nucleic acids also code for proteins, meaning that they have the instructions for all proteins embedded within that molecule. Now this is important because proteins are ultimately how we express our traits, from our eye color to the enzymes that our body uses to digest food. And there is a relationship between DNA and how we actually get to those traits that are expressed. So you might want to draw some of these pictures that you see over on the side, or at least make a little flow chart of how the information is organized. So we know from mitosis and meiosis that chromosomes contain DNA. Okay, that's what they're made of. And chromosomes are divided up into segments. Each segment of DNA that codes for a trait is called a gene. Make sure that you write down that word gene and know that it is a segment of DNA. And remember, DNA is made up of nucleotides. So the order or the sequence of nucleotides within that gene will determine the sequence or order of amino acids in the protein for which it codes. And ultimately, proteins allow us to express our traits. So make sure that you know from this slide, what does DNA stand for? What is its job? It carries our genetic material, it codes for proteins, and make sure that you understand how the information is organized. DNA is organized into chromosomes. Each chromosome has different segments. Those segments are called genes, and it's ultimately the nucleotides within that gene and that sequence of nucleotides that determine what protein is made by sequencing the amino acids of that protein. Okay, now let's talk a little bit more about the structure of DNA. Now, as I mentioned earlier, DNA is made up of nucleotides. Those are the monomers or building blocks of that molecule. So make sure that you know that DNA is made up of monomers called nucleotides. And each nucleotide has 3 parts. You may want to number those 3 parts. So you have a phosphate group, a 5-carbon sugar, and a nitrogenous base. Okay, so those are the 3 parts. So make sure that you know what are the 3 parts of a nucleotide. The phosphate group, the 5-carbon sugar, and the nitrogenous bases. So you've got your phosphate, a 5-carbon sugar, and a nitrogenous base. Now, as we progress through this unit, it's going to become really important that you know the names of the 5-carbon sugars that make up DNA and RNA, and the nitrogenous bases that can be found in each molecule. So make sure that you know that for DNA, its 5-carbon sugar is deoxyribose, and it contains, each nucleotide can have 1 of 4 bases. It can either have adenine, thymine, guanine, or cytosine. And from here on out, I'll use the initials that abbreviate them, so A, T, G's, and C's, or I may say their full name. So just if you hear A, that just means adenine, T means thymine, G means guanine, C means cytosine, and so forth. And those nitrogenous bases are split up into two groups based upon how many rings make up the molecule, either a purine or a pyrimidine. But it's not necessary that you know which ones are purines and which ones are pyrimidines. Now, on your paper, wherever you're taking notes, make sure that you draw a picture similar to this. So draw yourself a little circle to represent your phosphate group and put a P in the middle of it. Draw yourself a pentagon, little house type structure here to represent your 5-carbon sugar. And then for your nitrogenous base, you can just make a little rectangle and show them connected to each other. So you have a phosphate group, a 5-carbon sugar, and a nitrogenous base. And make sure you label those parts. So phosphate group, sugar, and nitrogenous base. You don't have to know the name of each nitrogenous base, just use the rectangle to represent it. Now on this next slide, it just kind of shows you the different shapes of the purines and the pyrimidines. Now you might notice something. I only mentioned 4 bases previously when I was talking about DNA. This 5th base here, uracil, is found only in RNA, so that's why I didn't mention it earlier. And we'll talk more about uracil as we get into RNA later. Now, we know from the video that we watched on Edpuzzle that DNA is a double-stranded molecule, okay, a double helix. So each molecule of DNA has 2 strands of nucleotides, and those 2 strands get connected together through something called hydrogen bonds. So they're connected to each other by hydrogen bonds. Make sure that you know that. We will ask you questions about that on the test. What type of bond holds those together? Now, these 2 strands of molecules or nucleotides that make up the DNA molecule are said to be complementary. So when we say something is complementary, complementary means they just go together. And there are certain ways that the nitrogenous bases of the nucleotides go together. And that was established from the experiments conducted by Chargaff, which was talked about in the Edpuzzle video. Now, Chargaff figured out that in any sample of DNA, the amount of A always equals the amount of T, so adenine is equal to thymine, and guanine is equal to cytosine. So that allowed Watson and Crick, when they built their model, to figure out how the bases would pair together on opposite sides of the DNA. And we can use that information that we've learned from Chargaff's rule to apply it to a scenario in which we have a known amount of DNA and we know the amount of one of the nitrogenous bases. So we're going to actually apply Chargaff's rule and do some math problems. So I'm sure your algebra teachers will thank me for keeping your math skills fresh. So if you would, on your paper, go ahead and copy down the Chargaff's rule example problem. You can pause your video, and then once you've written down the example problem, begin watching the video again. So hopefully you have already watched your written down the problem. Now we're going to work this out. Okay, so always start by writing down how much DNA is in your sample. So we're told that we have 200 micrograms total DNA. That amount has to be given to you to begin with the problem. And you also have to be told one of the 4 bases and its amount. So we're told in our problem that there are 75 micrograms of thymine. Okay. Now going back up here in our notes, notice this part right here. According to Chargaff's rule, if there's 75 micrograms of thymine, there should also be 75 micrograms of adenine because A is equal to T. So I'm going to also write down that there are 75 micrograms of adenine. So at this point, you want to add the 2 bases together. So when you add that together, you should get 155, or excuse me, 150. So you have 150 micrograms accounted for. Now, looking back at the total we began with, we started out with 200. We need to see how much is left over. So if you're thinking in your head, hey, I should probably subtract right now, you're right. So you're going to subtract the total minus the accounted for amount. So you're going to say 200 minus 150 in this problem, and that equals 50. And this is our remainder. So if you go back and think about DNA, DNA has 4 bases. So we have 2 more bases that we have to share the remaining amount with. So we're going to take the remainder and we're going to divide it by 2. So for our problem, that's going to be 50 divided by 2, and that equals 25. So that means there are 25 micrograms of cytosine and there are 25 micrograms of guanine in this sample. Okay, so there's 25 guanine and there's 25 cytosine. And if we go back up and look at our problem, we were asked how many micrograms of guanine is in the sample. So we've now figured that out. So 25 micrograms. Okay. So let's try another problem. So you can just write down these numbers that I'm giving you, and then you can work from there. So in our new problem, I want you to say that there are 300 total micrograms of DNA in a sample. And of those 300 total micrograms, let's say that 50 of them are cytosine. We want to know how many micrograms of adenine there will be. So thinking back to how I did the first problem, I want you to try now to figure this one out on your own. Pause the video, look at the steps that I have written out on the other side of the screen. When you're finished and think you have it solved, start the video again and check your work. Okay, so hopefully you've done it on your own now, and now you're checking your work. So we had 300 total. We have 50 micrograms of cytosine. So according to Chargaff's rule, you should also have 50 micrograms of guanine. So C and G are equal to each other. So if you add those two together, you should get 100 micrograms accounted for. Because remember, you added. So now you have to subtract. So that would say 300 minus 100, and that equals 200. So that's our remaining amount. And remember, your remaining amount is going to have to be divided by 2 or split to be shared equally among the remaining bases. So that means there will be 100 micrograms of thymine and 100 micrograms of adenine. All right, so hopefully that helps you figure out how to do those problems. If you need some more help with that, Miss Tippett or I will host live video feeds later this week, and you can ask us some questions. So now that we've practiced Chargaff's rule, and we know that Watson and Crick used Chargaff's rule to figure out how the DNA molecule would be held together, let's move on and talk about the actual shape of DNA. All right, so DNA is a double helix, and that double helix shape was determined by the work of Watson and Crick. So make sure you know that a DNA molecule is in the shape of a double helix, which means it has 2 strands that are twisted on each other, and that was established by Watson and Crick. And if you watched your Edpuzzle video, you know that they relied on the work of Chargaff to figure out the base pairing rules, and they relied on the work of Rosalind Franklin and Maurice Wilkins to figure out the shape from their X-ray crystallography. Now on the outside of that DNA molecule, the sugar and the phosphate groups are going to create what we call the backbone. So that's actually going to be on the outside. And then on the inside, you have the nitrogenous bases, remember, connected by the hydrogen bonding. So if you look at this picture up here, you're going to notice something, that they have these numbers, 5' and 3'. So that little mark just means prime. And those 5s and 3s are associated with which carbon they that are within the sugar. So this would be 1, 2, 3, 4, and then 5. Each angle represents one of the carbons. So this is the 5' end because the 5th carbon is kind of angled towards the outside. On this side, 1, 2, starting here at the point, 1, 2, and 3, the 3rd carbon is kind of pointing towards the outside. So the strands of DNA run in what we say are opposite directions. They are anti-parallel. Make sure that you know the word anti-parallel means that the strands of the DNA molecule run in opposite directions. One runs in the 5' to 3' direction, and we refer to that as the leading strand. And the other strand runs from the 3' to the 5' direction. We call that the lagging strand. And these terms, leading strand and lagging strand, refer to the speed at which those strands are replicated. So let's take a look at replication now. And again, replication speed depends upon which strand is being replicated and how quickly it can move through. Okay, so remember when we talked about the cell cycle and mitosis, which seems like a long time ago, DNA actually copies itself during S phase. So remember, DNA copying itself is called replication. And that happens during S phase, and that's happening inside the nucleus. Make sure you know where this is happening, and that should make sense because in a eukaryotic cell, the DNA is stored within the nucleus. And at the end of replication, you get 2 identical molecules of DNA. Now, Watson and Crick, when they built their molecule in the early 1950s, they actually predicted how DNA would replicate. And later scientists confirmed this. And they said that each strand of the DNA molecule could serve as a template or a pattern from which a new strand could be built using those base pairing rules that were established by Chargaff, A to T, G to C. Now, as the process of DNA replication was confirmed, scientists came up with a term to describe that process, and they said that replication is semi-conservative. And if you break down that word, you'll notice that semi means half, conserve means to save. So essentially, as DNA is replicated, each molecule produced contains half of the old DNA and half of the new DNA. So half old or original and half new. So each molecule has one original strand and one new strand. So semi-conservative. One old and one new strand. All right, so what do I mean by that? So on your piece of notebook paper, wherever you're taking your notes, I want you to copy this down right now. And you're going to need 2 different colors. So one color that you use will represent your original DNA, and one color that you use will represent your new DNA. So pause the video, copy this down, and when you're ready to proceed, restart the video. Okay, so as I said earlier, DNA replication is semi-conservative. So when we say that replication is semi-conservative, we mean that each old strand or original strand will serve as a template to build the new strand. So if you look here, this was my original DNA molecule, and using the base pairing rules, you can see how they pair together. So on one side of the molecule, there's A for adenine. On the other side, there's T for thymine. Then you have G for guanine, C for cytosine. T for thymine, A for adenine. C for cytosine, G for guanine. During replication, the the molecule will uncoil out of its helix and it straightens out kind of like a ladder. Then those two sides are going to split or separate from each other, as shown with these arrows. Now, each strand of nucleotides is now a template to build a new strand. So these are our old DNA strands, and then we're going to build the new ones using the base pairing rules. So if there is an A for adenine here, what should pair up with it? Hopefully you said T. So A pairs with T. Okay. All right. Moving on, G would pair with C. T would pair with A. And C would pair with G. Now over here, we get the same process happening. Here's a T. So on the new strand, there should be an A. All right, here, there's a C. So on the new strand, there should be a G. On the old strand, there's an A. So on the new strand, there should be a T. And on the old strand, there's a C. So on the new strand, there should be a C. So this process, remember, is said to be semi-conservative, half old, half new. So on the outside of your molecule, right, sorry about that, old, and on the inside put new. So half old and half new. So semi-conservative, half old, half new. Make sure that you know your base pairing rules. So A always pairs with T, G always pairs with C. All right, so this diagram here just kind of shows you the process of replication. Old strand uncoils, opens up, new bases are added, so you get half old and half new DNA. Semi-conservative. Okay. So now let's talk a little bit about the steps of replication. So in order for the DNA to actually be replicated, the DNA has to be unwound, okay, or straightened out. And the strands are separated. So they unwind and straighten. So if you think about your arms being like twisted together, untwist them and then separate them. This happens by an enzyme called DNA helicase. All right. If you are in honors biology, you need to know what each enzyme does. If you're in regular biology, you just have to know what's the first step. The DNA is unwound and the strands separate. Okay. Now, there are some proteins that once those strands have separated are going to bind onto those single-stranded DNA sections and prevent them from joining back together. You don't necessarily have to know the names of those. Okay, so now that we have the DNA open, okay, and where it opens up on that molecule is called a replication fork, new nucleotides are going to be added onto the DNA molecule. And those are added on by something called DNA polymerase. So make sure you know free nucleotides are added to the exposed strands, okay, and that's carried out by an enzyme called DNA polymerase. Now, make sure that you know that the speed of replication differs based upon which strand is being replicated. So when a leading strand is being built, it's being built in the 5-3 direction, and it occurs continuously. So it's going to be fast. Lagging strand, in contrast, is built on the strand that's going in the 3 to 5 prime direction, and it's built discontinuously. So what do I mean by discontinuously? Essentially, it's built in segments, right? So the replication fork opens up, it adds some nucleotides, then it has to wait. Okay, the enzyme moves, it builds another segment, and then eventually those segments get put back together. And those segments are called Okazaki fragments, named in honor of the Japanese scientist who figured out how leading versus lagging strand replication occurs. And eventually, those segments are put together by an enzyme called ligase. And after that, you have your two new molecules. They're going to basically twist back into a helix, and each one's going to have half old DNA and half new DNA. And remember that the word for that is semi-conservative. Now, once you've done your notes, I want you to watch a tutorial video that animates the process of replication for you. So make sure that you take a look at that. I think it will help you understand the process a little bit better. And this is just a picture showing you how the replication fork opens up and how on the leading strand that moves from 5 to 3, notice that there are no segmentations. And here on the lagging strand where it goes from 3 to 5, it will build a segment, then it waits, build a segment, then waits, build a segment, and wait. And those little segments get put together, and remember those segments are called Okazaki fragments. Okay. Hopefully you have understood most of this video. Again, Miss Tippett and I will be offering live sessions for you guys to help you in the future. Please join us for those to help answer questions. Hope you guys are doing well.